Paula Bernardo, and I will talk about new synthetic compounds for glioblastoma treatment. So, gliomas are brain tumors derived from glial cells, and they represent around 40% of central nervous system tumors. And they are classified by the World Health Organization in 4 grades of malignancy. Grade 1 is considered a benign tumor, and the other grades, as higher the grade, higher the malignancy of these tumors. And glioblastoma is the highest grade and the most malignant one. So, in the focus of our study. So, as I said, glioblastoma is the most frequent and aggressive glioma, and the mean overall survival is only about 12 months. The treatment is based first on surgery, and we know that the surgery has a prognostic value, so the higher the extension of the resection of the tumor, higher and bigger is and better is the prognosis of the patient. And it's followed by a concomitant treatment of chemotherapy associated with radiotherapy, followed by adjuvant chemotherapy. The first-line chemotherapy is temozolomide. There is an alkylating agent, and it increases overall survival in about 2.5 months. And it looks short period, but as was said before, it has a great improvement in quality of life, and it's less toxic than the previous treatment option, and that's why it's now the first-line chemotherapy, although it has...
Yeah, I just add one word on temozolomide, also talking about what we discussed this morning. Temozolomide is a nitrosourea, which comes from the mustard gases. So it's a family of compounds that is old to very old. Temozolomide was first explored in melanoma, and it's through the UK Cancer Research and the company in UK that they targeted it in glioblastoma, because also glioblastoma and melanoma have the origin of cells are relatively similar. What I want to mean is that we've been talking of new medications for glioblastoma, meaning that this is a place where there is a great need for new drugs and new compounds with modes of actions.
Yeah, and then because the gliomagenesis isn't well understood, so there isn't, now they are raising some molecular potential targets such as EDH1 that can explain a little bit more about the gliomagenesis, but as it's unknown, it's hard to find therapeutical targets. So there is few molecular targets raising in glioblastoma, because we are still trying to understand how the disease is originated. And then, it's needed because all this short survival and the short known about the disease progression, new synthetic compounds has emerged and are important for treatment of this disease. And our group, associated in collaboration with 2 other groups in Brazil, are trying to develop new therapeutical targets, not only for glioblastoma but our cancer types. And there are 2 new compounds named LQB-118 and LQB-223 that has potential activity, mainly against myeloid leukemia cell lines. So by now we know that LQB-118 has a potent effect against myeloid leukemia cell lines, reducing its viability by production of reactive oxygen species, and also inhibiting inhibitor of apoptosis proteins like XIAP and survivin. And they can also modulate and inhibit the expression of several transcriptional factors such as FOXO3A, FOXO-M1, and NF-kappa-B, and all of this inducing apoptosis. But little is known about the role of this compound in solid tumors. And there is only one study in literature showing that it has a potent effect against prostate cancer cell lines, reducing viability and inducing apoptosis by inducing expression of P21 and inhibiting MYC and cyclin D1. So, and it's also important to know that this compound has low toxicity to bone marrow of mice. So our group demonstrated that there is no toxicity for bone marrow cells and for bone marrow population, subpopulation of cells, and it's also effective against patient samples with acute myeloid leukemia, demonstrating that it's a potent antitumor agent and with low toxicity. On the other hand, the LQB-223, there is only one study demonstrating that it reduces cellular viability, promotes cell cycle arrest, and induces DNA fragmentation in a leukemia model that is resistant to different drugs. It's a multi-drug resistant model. So our goals are to evaluate the tumoral activity of these new synthetic compounds in glioblastoma cell lines and to gain better understanding of the pathways underlying the compound's mechanism of action. So first, to evaluate the antitumor activity, we used the 3 human glioblastoma cell lines, named U251, A172, and T98G, that they have P10 mutation, and the U251 and T98 has TP53 mutation, and the A172 has TP53 wild type expression. So first we evaluate the cellular viability by the MTT assay, and we observed that the LQB-118 compound reduces cell viability with a low concentration of 6 micromolar, and has significant effect since 24 hours, and in 72 hours it reduced almost 90% of the cellular viability. And the other compound has a significant effect only after 72 hours in the glioblastoma cell line models. So we also demonstrated that they can induce cell detachment from culture flasks as they grow as monolayers. When they detach from the culture flasks, it means that it's a signal of cell death induction. So we can see that the LQB-118 induces all the cell line cell detachment, and also the LQB-223. So, and it's important to know that they had a greater effect than the actual treatment, temozolomide, as it does not induce cell viability reduction. It doesn't induce cell death by trypan exclusion assay, and it does not induce cell detachment from the culture flasks, suggesting that these new compounds has a better effect in the cell lines than the first line treatment option. So we see that it has an effect in the cellular proliferation and viability, but is it inducing cell death? So we evaluated apoptotic cell death, and we could see that temozolomide in these cell lines couldn't induce apoptotic cell death, not by annexing labeling, not by reduction of procaspase species, procaspase 7 expression, but the LQB-118, the higher concentration of 12 micromolar, could induce almost 60% of annexing labeling and reduce the procaspase 7 expression, suggesting an activation of this caspase. And the LQB-223 also induced annexing labeling in a smaller extension than the LQB-118, but it also induced apoptosis and also promoted the caspase 3 activation. So to better understand how this is happening, we decided to evaluate survival signaling pathways, as they are important regulators of cell death and cell proliferation. So the MAP kinase pathway is activated by a tyrosine receptor that, once activated, are going to trigger a cascade of tyrosine kinases that culminate with ERK activation, that's going to activate transcriptional factors responsible for apoptosis inhibition, cellular proliferation, and cellular survival. And as you have heard on the previous presentations, the microRNAs has emerged recently as important regulators of proteins and signaling pathways. And the microRNA 7 is an important inhibitor of the signaling pathway by inhibiting RAS expression and ERK phosphorylation. So microRNAs are small non-coding RNAs that they are translated by RNA polymerase. They are processed by many endonucleases, and they form a single strand mature RNA that are going to target the messenger RNA. If there is a perfect complementarity to the messenger RNA, they are going to cleave the RNA. if there is not a perfect, it's just going to repress the translation. What means to inhibit the protein formation. So it can, at the end, inhibit the expression of many proteins inside of the cell, as it has many different, several targets. So to better understand the LQB-223 mechanism of action, first we decided to address if it, as was observed for leukemia cell lines, if it was also modulating the cell cycle. And we also demonstrated that it induced G2M arrest that is followed for enhancement of the DNA fragmentation and a loss of the profile of cell cycle. Furthermore, in demonstrating the cytotoxic effect of this compound. We also evaluate the microRNA 7 expression, and we could see that there were enhancement of this expression in U251 and the T98 cell lines that were in accordance with a reduction of RAS expression and ERK phosphorylation, suggesting an important role of this signaling pathway in response to this compound. The A172 cell line didn't show the same profile, but as we have seen before for leukemia cell lines, sometimes the same compound has different ways of action depending on the molecular markers of the cell line. And as this cell line has a difference in TP53 status, maybe it can be involved with the different cellular response to this compound. So about the LQB-118, we also evaluated the signaling pathway, as we have previously known that this compound regulates NF-kappa B expression, many other transcriptional factors that are activated for this pathway, and also inhibitor of apoptosis proteins. So we decided to look up which signaling pathways can regulate all of these targets of the compound. And we have recently demonstrated that the LQB-118 can modulate the NF-kappa B pathway through alteration of microRNA's expression. So we are going to evaluate the microRNA 7 expression and also the microRNA 210. There is an important for solid tumors, as it responds for hypoxia, and it modulates mitochondrial metabolism and cell division, although its role in apoptosis and cellular proliferation is still controversial in literature. We know by now that these microRNAs regulate mitosis-related genes, impairing mitotic progression. It can inhibit anti-apoptotic proteins such as BCL2 and induce caspase 7 activation. And a natural compound was able to inhibit tumor cell growth by inducing this expression. So we know that in glioblastoma it's not well understood how does it work, because some studies demonstrate that it can be related to tumor progression, but we don't know if it's a consequence of a cause of tumor progression. So we demonstrate that the LQB-118 can reduce ERK phosphorylation and also induce microRNA 210 expression. And it does not modulate microRNA 7 expression, demonstrate a different profile of observed with the other compound, and the temozolomide cannot modify the expression of these microRNAs and cannot alter the phosphorylation of ERK protein. So, as I stated since the beginning, the major treatment option is the combination of radiotherapy with chemotherapy. So we try to address if we use a lower concentration of our compound associated with radiation therapy, if you would have additive or a synergic effect. And we use 3 different approaches. First, we treated with LQB or TMZ, and after with radiation, or first with radiation and after with the compounds, or the associated treatment. And we observed that the treatments alone, the black bars, did not have a cytotoxic effect, but when it was added, the dark gray bar, with first radiation therapy, it has no effect as well. But when we use the compounds first, the light gray bar, or at the same time, the white bar, we can observe that the LQB-118 has a synergistic effect, while the temozolomide has an additive effect, suggesting that when you use a lower dose of our compound with radiation therapy, we can have a synergistic reduction in cellular viability by triathlon exclusion as seen.
But
So,
just as you come back to the slide.
yeah.
So the white bars, it's concomitant treatments that you irradiate and you expose to the drug at the same time.
Almost the same time.
And the other one is you first ionize.
The first ionization is the dark gray bar, that there is no effect.
Yeah, that's
Demonstrate that it's important, or you give your chemotherapy first or at the same time with the radiation, because when you use the radiation first, it doesn't have the same effect.
In other words, you have the impression that you sensitize the cells to radiation.
To radiation when you treat first with the chemotherapy. At least in cell lines, I don't know if it's really different.
And that desensitization is much more if you put them all together at the same time.
Yeah, the best results are when you put all together. That's what is the pattern. It's the treatment actually is both together and after the adjuvant with chemotherapy.
But it's on cell lines in a dish, and you keep them for one day, but you don't wash them.
No, we don't wash them because of the radiation effect.
No, but I mean, if you give the drug first, they've not washed the drug before the radiation.
No, I don't wash the drug. So, in summary, we know that the compounds can inhibit cellular viability, induce apoptosis, and inhibit the ERK pathway, but how? For the LQB-223, we believe that it's mediated by microRNA7 expression that inhibits RAS and ERK phosphorylation, inducing apoptosis. And for LQB-118, the microRNA 220 may be involved as it plays a role in the mitochondrial metabolism, what would explain the high reduction in cellular viability observed. It also can activate caspase 7 as we observed for the treatment with the compound. And we don't know yet how is it regulating ERK phosphorylation as microRNA 7 was not involved. So, and finally, when we associate it to radiation, we have a synergistic effect. So, taken together, our results suggest that LQB-118 and the LQB-223 are promising anti-tumor compounds with distinct mechanism of action in which microRNAs plays a role. And the perspectives are really understand the role of these microRNAs by modulating their expression and then being really address how they are working in response to these compounds. So I'd like to thank my supervisors, Raquel, Maya, and Leila, from the Brain Institute and from the National Cancer Institutes, the biochemistry laboratory that synthesizes the molecules, the founding agents, and the scientific committee of this conference for the opportunity for presenting my work.
What's the specialty of the baby?
What?
What the specialty of the baby is?
I don't know yet. We don't know. But it's my lab group in Christmas meeting and confraternization.
Thanks a lot. Presentation. Questions from the audience? Maybe not right now, maybe after, I don't know. Yes?
So, in your last result, it was very impressive. So the synergistic effect with irradiation. So this result suggests that your compounds affect somehow the checkpoint, DNA damage checkpoint. So have you ever considered the effect on the ATM, ATR, or checkpoint kinase pathway?
Yeah, I think about this ATM pathway, but we haven't addressed it yet. But it's really an idea, but I don't know if it's working in the DNA damage pathway as temozolomide works similar like this and doesn't has effect in our cell lines. So I don't know if it's gonna be effective in these pathways, but we have already thought about it, and we want also to address, make more really cell death studies to confirm this synergic effect and try to understand how is it happening, because we know that like LQB-118 can also target topoisomerase alpha. But in biochemistry assays, we are going to address it now in biology, in the cell line models, and maybe it can be a way that it's altering the DNA damage or inducing DNA damage as well.
I have a couple of questions. What does LQB stand for? What is the meaning of LQB?
It's a laboratory of chemistry, bioorganic chemistry, but it's in Portuguese.
Okay. And these molecules are patent protected?
Yeah, the LQB-118 is patent protected.
And the other one?
The other one, no, I think they published it before any patent. Yeah, so it's public domain right now.
Did you try to put all together radiation, LQB, and chemotherapy?
No, I haven't tried it. Yeah, no, I tried all of them.
It might be dramatic, I don't know.
Yeah, maybe.
Okay.
And also try the LQB with temozolomide. Maybe that has some different effects.
That would be interesting. Did you investigate, currently there is quite a lot of trends in stem cells, especially in glioblastoma. Because it's more certain that stem cells, well, the cancer-initiating CICs are responsible for the relapses of the glioblastoma, which is what in the end kills the person. With the surgery, radiotherapy, the work is more. Did you investigate? Did you have the opportunity to investigate with the CICs?
So we tried once to do it, but they are really in small proportion, about 1%.
1 to 5%.
So in our cell lines, we're about 1%, and then we tried first with cell sorting, but it would take hours to get. And we tried with the magnetic columns and beads, but we couldn't have really accurate fraction. So by now we didn't try it because we couldn't really do find a protocol that we could have a fraction pure and with cells to try it.
Okay, it would be interesting. Well, which cell line are you using? Is it your own cell line or is it the classical cell line?
It's the most results are with the U251, that it's highly, it's the most used one, and the other used one, but we don't have it, is the U87, that it's also really studied.
Which is coming from Geneva. What are the regarding the molecules themselves? Besides the research, there are some perspectives of development towards a drug, towards a therapy, or whether you stand with that?
These drugs, I think the most advanced, you can say, one is the LQB-118 that has already mice studies with melanoma cells, and they demonstrated the reduction of tumor volume and tumor weight, and they treated healthy mice with this compound, and they had no side effects and no alterations in bone marrow. So there are more results with this compound than with the other one that was synthesized after.
Yeah.
So I think they are the most promising one, but there are more results also with melanoma and with leukemia models. And we are there because the lab that might make the mice models are a different one. We are 3 labs. The lab I am is working with the in vitro part, and there is the chemistry lab that is producing the molecules and changing its structure, and there is another lab that making the in vivo part.
So it's soluble and it goes through the very body?
That's the question that everybody makes. I don't know it yet. Although there are some controversies that many glioblastoma patients, the brain blood barrier is already broken and disrupted.
That's very true. That's very true.
So don't know if it's, but we want to address it, and there are now strategies such as nanoparticles and some kind of chips that you can insert in the tumor. So there are ways to deliver this.
Or by
this drug
chemistry
to-
and radioactive
Yeah.
C14, C13.
There are other strategies to deliver it to the tumor.
Okay.